Okay, so my name is Athi. I am from IIT Madras, and I'm glad that I was invited to this very interesting conference. I see some familiar faces as well now. And the thing is, I think I was here about 24 years ago where we had this National Science Day as part of the Anna University Center for Biotechnology, where we actually walked into this auditorium is what I believe. And maybe it's quite revamped, but it kind of gives me some goosebumps a bit. Right, so but it's nice to be here after so many years. It's first time I'm speaking here at IMSC, and the talk, I thought I will kind of structure it in a way where the audience here can understand. So I thought I would include something on phase separation and oligomerization and how these could be linked. Right. So before getting into phase separation and oligomerization, I would like to pose a general question, which is something that we are very interested in the lab is. How do bacteria sense environmental cues, right? So you have bacteria on the soil and it need not express all its genes, right? For example, toxin genes or any of these things, right? Because it need not have these particular proteins going inside the cell and not being able to do anything. So it needs to sense the environment, and whenever the environment is favorable, say for infection, then it can express that particular toxin, and then the toxin can have its effect. So how does bacteria sense this? And bacteria and many systems for that matter are filled with sensors. And all of these proteins that you have, even membrane, RNA, all of these are effectively sensors. So you can actually think of two kinds of sensing mechanisms in a very broad term. One is non-protein based sensing, in the sense that when you have changes in environmental conditions, the topology of DNA or the plasmid, whatever you have inside the bacterial cell can vary. The RNA structures can vary. Maybe this can be transcribed, this cannot be, this can be translated, and this will not be possible to be translated. You can also change the fluidity of the membranes, and that is another possibility in which it can control the extent to which things can move in and out, and hence sense the environment. But what we are interested in is protein-based sensing. When you have small changes in temperature going from say 25 to 37 degrees Celsius, or changes in osmolarity or the salt concentration within inside and outside the cells, you can have these transitions going from a compact globular structure, as what Monica was talking about in this case with respect to proteins, to disordered like structures, or you can have disassembly of oligomeric systems, and you can also have disassembly of homo-oligomers or disassembly of hetero-oligomers. Then when one of these happens, the bacteria is able to kind of switch the genes. Some of these can bind DNA, and this might not be able to bind DNA, hence repression can happen or activation can happen, and this on-off state can be tuned. I believe there's some issue with these slides, but anyway. So bacteria in general are sense many different metabolites, and not just pH and temperature, but even antibiotics and different kinds of substrates that they have in the environment. Right, so and particularly it's more interesting when it comes to multi-drug resistance, because bacteria also kind of exchange genomic material across them. So it will be interesting to figure out what are these sensing mechanisms bacteria employ to actually sense environment and protect the DNA. For example, what happens is that when the environment that the bacteria grows is not very conducive for its replication, it tries to protect its DNA from external agents. It kind of condenses the entire nucleoli, similar to what we heard in the first talk on the nucleosomes. Similarly, in bacteria, you have systems or nuclear condensing proteins that condense the DNA such that there are no external agents can act on it. And we are interested in these kind of, because of the switch from Mac to this, there's some issue with the placement of these holders. So in bacterial systems, how does this DNA get condensed? And that's what we are interested in. And these particular systems that condense DNA are very sensitive to temperature and environmental osmolarity. And these proteins are called HNS-like proteins, or HNS family of proteins. And these are similar to histones in some way, but these are non-specific in the sense that they bind non-specifically to DNA and kind of condense the genome or the DNA. So what is known so far, and this is based on many different experiments, biochemical experiments, is the fact that at low temperature or at low salt concentrations, as I call it ion extent here, these protein molecules are able to oligomerize, assemble on the DNA, and kind of bring these guys together. Right. So, and because of this, these genes that are present here are unable to be transcribed, and hence there is no activation, or the gene is not in the on state, and hence there won't be any expression of a gene. But at a higher temperature, say, body temperature of a human host, particularly for enterobacteria that infect our gut. When the temperature is constant, which is a very interesting or a good thermodynamic variable if you can think about it, or high ionic strength, which is what happens in your gut, what happens is because of this small shift in temperature, it's just about 10 Kelvin or 10 degrees Celsius in this particular case, these guys are disassemble, and when they disassemble, what happens is that these gene sequences that you see here are available for further transcription and hence translation, and you can have some kind of expression that can take place. And this is the overall mechanism, but the question that has not been answered so far is how exactly does this happen? How does this switch happen? And that's what we are interested in addressing. So this particular protein HNS, which I have shown these in circles out here, this is a monomeric form and this is the oligomeric form. I'll show or talk more of it in the next few slides. And it's involved in many different functions within the bacterial cell. It's actually a multifaceted system. It is involved in genomic compaction. It prevents maybe sometimes transcription, even enables transcription. It is involved in the silencing of foreign DNA, that is a DNA which is not native to that particular bacteria. It is also involved in expression of certain virulence genes. It controls nearly 5% of the open reading frames or genes that can be transcribed in bacteria. And it's also implicated in many multi-drug resistance systems, right? So what we're interested in is how does HNS act as a biosensor and hence can be implicated in bacterial survival and virulence. And of course, this is a system that has not been worked on in great detail, which means if you can come up with a drug molecule that acts against this particular system, which is not very well studied, you can have a novel class of antibiotics that one can come up with, right? So, and what I'm going to talk about in this particular section is all the structural organization and assembly modes of this HNS. And so just to highlight, this is the actual structure of the monomeric form of this particular protein HNS. So you have something, this is the structure that is predicted by AlphaFold. The entire structure has not been solved. This is the AlphaFold predicted structure. And this is the cartoon of this particular structure that I'm showing you here. And effectively what you have is something called the dimerization site. You have an oligomerization site, and you also have something called the DNA binding domain. This is the domain that binds DNA. And these two, this site oligomerizes with or dimerizes with another such monomer, and hence can form a dimer. As you can see here, these yellow guys have come together and they can form a nice little dimer. But now you can see that this oligomeric sites are also available for a higher order oligomerization, and you can actually form from a dimer to a tetramer to an octamer to a 16-mer and so on and so forth. Actually, it can go quite large. But of course, we do not know what exactly is the true size of this particular system. And this is what we set out to study, the true oligomeric nature of this particular HNS molecule. And what we did was we did something called the size exclusion chromatography, which is a very simple technique. What you have is or something called a gel permeation chromatography. This is something that you might be familiar with. You pass a whole bunch of material through a column which has multiple pore sizes. The larger molecules will come out first, and the smaller molecules will get trapped within these pores and come out later. And what I'm showing you here is the elution profile at some high ionic strength condition, because that's where you can run this particular experiments, but at low protein concentration, which is around 15 micromolar. What you can see is this is about the maximal elution volume that you get here, the peak. But as you increase the concentration of the protein, what you can see is that the protein comes out earlier and earlier, meaning the elution volume becomes lower. As you can see, a left shift here. So which means that at 200 micromolar, it elutes at around this particular volume on average, while at 15 micromolar, it comes out much later, which means there is a concentration dependent assembly of this particular protein. Of course, this tells you that there is some kind of concentration dependence in terms of the size of particles that you have in the solution, but it doesn't tell you what are these different oligomeric states of this particular protein. So we did something called analytical ultracentrifugation, and this analytical ultracentrifugation is a very simple technique. What you do is you take a protein solution and you kind of monitor when you spin it at high speed what happens to the so-called sedimentation front, and you can fit this to a modified version of the fixed second law, which is called the Lamb's equation, and you can get information on the diffusion coefficient, Stokes radius, and so on and so forth. And also something called the sedimentation velocity, and that's what we have here. So what you have here is some sedimentation coefficient, which is a measure of the size of this particular particle, and this is the distribution. So when we did this at low concentration, at low temperature, and at low ionic strength condition, which is about 150 millimolar, and at 15 micromolar, and what you can see here, there are three distinct peaks that turn up. And this corresponds effectively, you can actually model, take the structure, the alpha-fold predicted structure, and you can actually calculate what the hydrodynamic radius is, and you can actually even predict the sedimentation coefficient. And when you do that, you find that this is likely the monomer, and this is the dimer, and this is the tetramer. So the protein HNS in its native state and at low concentration is in an equilibrium between 4 or 3 different forms in this particular case, which is effectively a monomer, dimer, and a tetramer. It is not just a monomer in solution. But now as you increase the concentration to 15 or 125 micromolar, what you see is that there is some shift in the profiles. I would not have time to discuss why that happens, but what we identify is that there is not just a monomer, but also a dimer, tetramer, and also an octamer that is present in solution. All these are freely exchanging in the solution, right? So there is a monomer, dimer, tetramer, and an octamer. And we also did additional assays to confirm this, and again, I would not get into this. And to conclude this part, what we see here is that the concentration-dependent population distributions of higher order molecular assemblies are identified, and there is an equilibrium between a monomer, a dimer, tetramer, and octamer, which I am showing you here. This is the monomer, dimer, tetramer, and octamer. This is a picture we will be using quite a bit. And we also see that when you change temperature, there is a change in the helicity of this particular protein. What I am showing you here on the x-axis is the temperature, and the y-axis the helical structure, the extent to which there is a helical structure present. As you go from low temperature to higher temperature, the intensity of this particular signal decreases, indicating that at some normal growth temperature of bacteria, optimal growth temperature of a bacteria, or the body temperature of a human host, there is a loss in helicity as well. So when you have HNS, its conformation, meaning the kind of states that it can populate, not only depends on temperature, but also depends on the concentration of the material that you have present here. All right. So now that you have this particular variation, and now the question is, now let's say you have a bacterial nucleoid where you have the proteins that have already condensed on the DNA. Now if the bacteria finds itself in a favorable position, now how does this bacteria regulate itself and says, hey, I am in a nice position right now, I want to express all the genes and hence infect the human host. So how does it know this? So how does it sense this, or how does it dysregulate or disassemble this particular oligomeric structure that it has formed? So this is something that we had in mind, and when we looked into the structure, what we found was this in this oligomerization site, not the dimerization site, but in oligomerization site, what we found was that some charge-charge interactions between the aspartate, lysine, and this lysine that is present here. This is one protomer, as we call it, or one monomer, and this is the other monomer that is shown here. You can see there is charge-charge interactions that are present across the chains. So, and what we found was this tyrosine in bacteria actually gets phosphorylated. And this particular phosphorylation, we hypothesized that this phosphorylation results in long-range repulsion between the negative charges that will be present on this particular phosphorylated tyrosine and the aspartate that is present here, and hence this guy will actually disassemble, meaning it will lose its oligomeric status, but it will still be a dimer. It will not be able to form tetramer and octamer, but it will be still able to form a dimer. And this is our hypothesis. We actually did a series of experiments. I will not go into this. But when we did the same elution profile, which is a size exclusion elution profile of this particular protein, what we saw, unlike the earlier distribution that we saw for HNS, which has this broad elution profile and this peak that is shifted to about 160 ml, what you can see is that when you have this Y61E variant, meaning you take this mutant and you express and then you purify and you pass this through the column, what we see is that it's a very sharp elution profile, which is what you would expect it for a well-folded system, which does not have too many different states. And if I can go back, when I change the concentration, the maximum or the peak position does not change, meaning the particle size that is present in solution does not change. And this is just a representation of the same thing. And when we did analytical ultracentrifugation on top of this, and what we found was that all we found was a single peak, one that is corresponding to that of a dimer. So which means that when the phosphorylation happens on this particular system, you just have a dimer. There is no monomer that was present before, and there is no tetramer, and there is no octamer that is present. So the single phosphorylation event is able to switch this protein from being polydispersed to being monodispersed, having a single molecular species in solution, which is actually completely dimeric. Again, I will not go into that particular representation, right? So now you have this particular information, and we can see very clearly with concentration that the oligomerization status of this particular protein varies. The oligomerization status also varies with the temperature, but we cannot do this analytical ultracentrifugation studies at very high temperatures. We cannot do it at, say, 280 Kelvin, we cannot do it at 310 Kelvin. And if you really want to do it at 310 Kelvin, that's what you really need to study, because that's the growth temperature of optimal growth temperature of E. coli particularly. So, but to kind of get a handle on what exactly is the oligomeric status on this protein, we did something called thermodynamic modeling, which is quite simple. And to do thermodynamic modeling, we need to have some information on the temperature dependent population of these different species, and that is something very difficult, but one can do something called differential scanning calorimetry, which is effectively the measure of the heat capacity of the solution as a function of temperature of this particular protein. And what you have here, this is noise, but what I'm showing you here is at 15 micromolar and at 150 millimolar ionic strength condition, you have a transition which has a peak at around this particular temperature, and there is a shoulder, and then there is basically flattens out. But now as I increase the concentration of the protein, the peak increases, because there is a larger enthalpy that is required for the larger particle to actually unfold, because at higher concentration you have larger sizes, and hence you have a higher enthalpy, and you can see that the peak has shifted to the right. And you use even higher concentration, the enthalpy increases, and hence the amplitude of the CP peak increases, and it's even further shifted to the right, and you can still do it at higher concentration, and it's all shifted. But the interesting thing is that you see that there is a shoulder here which is not present in the last two higher concentrations, and this gives you some indication as to what kind of assembly modes are present in the particular system, right? So if I take a moment, and if I can go back very quickly. Right. So if you see this, and we label this as a monomer, this is a dimer, this is a tetramer, and this is an octamer. But you can see that if the system is able to form all kinds of oligomeric species, which means monomer, dimer, trimer, tetramer, pentamer, hexamer, and so on and so forth, you will have a continuous distribution of these particular sedimentation velocity profiles, or the sedimentation coefficients, and we don't see here. So all we see are discrete profiles. And because of the fact that we see discrete profiles, and because that we can use the structure and hence model these and kind of assign these, we kind of come up with a very simple logic to actually capture both the concentration and temperature dependent data, which is what I'll be talking about here. So the idea here is that there is an octamer that can dissociate to form a tetramer with an oligomerization constant, which is Ko. This tetramer can dissociate to form two dimers, and the dimer can dissociate to form a monomer. This will be the Kd, which is a dissociation for a dimer, and this will be Ko, which will be the dissociation for the oligomer, and the monomer can be also in an equilibrium with an unfolded state. This will be represented by this equilibrium constant, that is Ku. And hence, to completely capture the features of this particular system, you will need Ku, Kd, Ko, and also the enthalpy of associated with this particular transitions, and hence the entropy as well. So this is the simplistic representation that you can have here. N8, which is the octamer, forms N4, and the octamer, the tetramer can form a dimer can form a monomer, and the monomer is in equilibrium with an unfolded state. I can write this in the form of these equilibrium constants, and I can also assign very specific enthalpy changes to this particular processes, each of these process X being O, D, or U, which is for oligomerization, dimerization, or unfolding. And I can basically do a simple mass balance and rewrite this particular expressions in terms of the equilibrium constants, and calculate the total partition function and also the partial partition functions, and I can have the temperature dependent partition function of this particular system. And once you have this, you can actually calculate the heat capacity profile by taking derivatives of this particular partition function, and hence I can actually reproduce the experimental heat capacity profile at a semi-quantitative level, not at a perfect quantitative level. And because this will give you information on what kind of shapes that you will obtain, and if you have heat capacity profiles that you can obtain, then you can actually calculate the fraction of these different species. And we know already from the analytical ultracentrifugation studies that you know what are these different species that are present and the fractions of each of those particular species, right? So, and what we did was, when we used a certain combination of parameters, what we were able to reproduce is a trend which is very similar to what I have shown you before. At low concentration, you have two distributions with the shoulder here, and as you increase the concentration, this shoulder disappears and all of these kind of merge exactly as what we saw in the heat capacity profile before. And when we looked into the dimerization constant and the oligomerization constant, what we found was that the dimerization constant is extremely strong, meaning the dimer that is formed is quite stable, and the dimer effectively seeds higher order oligomer formation, and you can see that the oligomer has a very weak association constant that is shown here, and very independent of temperature between 298 and 310 Kelvin, but the dimerization has a very steep temperature dependence. So effectively, the story here is that the dimer seeds the self-association into higher order oligomers, and again, I would not get into this because of these issues here. But since you have this particular model, and since this reproduces the populations, I can actually plot the population of these different species as a function of temperature. And what you can see here is that as a function of temperature, the octamer at low concentration is very low, and the tetramer is on the higher side, the dimer is even more so, and the dimer and monomer are the most populated states for this particular protein at this particular concentration, and that 150 millimolar Anixton condition. But when you increase the concentration, what you can see here is that at about 300 Kelvin or about 310 Kelvin, you can have all kinds of species that are present. And this kind of explains some of the observations in literature where anyone who worked on this particular protein, everyone was giving a slightly different answer as to what is the true oligomerization status of this particular protein. Some were saying octamer, some were saying it's a dimer, some were saying it's a monomer, but now what we identify is that you have a mixture of all of these states in solution, and that's what we were able to identify, and we were actually able to reproduce the experimental populations from analytical ultracentrifugation quite well, right? So we also found that we did the same experiments at higher Anixton condition, and whatever I talked before is at lower Anixton condition. When you do it at higher Anixton, which is at 300 millimolar, which is a shock that bacteria generally face. And what we found, surprisingly, despite the kind of oligomerization we see here, we still see the same oligomerization status, monomer, dimer, and a tetramer, and at the higher concentration still a monomer, but these peaks are on the lower side, which means the dimer population is reduced and the tetramer population is reduced. At the expense of these, the octamer population is actually significantly enhanced quite a bit. And there seems to be kind of a difference in the assembly cooperativity when you change the Anixton conditions. And we also were able to do the heat capacity measurements on this, and we were able to actually model this very well. And in this particular case, what we found was that the dimerization constant is weakened quite a bit compared to the oligomerization, and hence you have all of these melting simultaneously here, while in the case of lower Anixton condition, you have decoupled transitions because you had these two peaks that we were able to identify. Right, so at higher osmolarity, Kd becomes very similar to the Ko, and hence there is also a weaker dimerization that happens. And the oligomerization becomes slightly stronger, as I'm showing you in reference to the 150 millimolar Anixton data. So effectively, when you change Anixton conditions, the bacteria is able to somehow sense this because there is an interplay of these particular oligomerization constants that take place, right? So what has been known also is the fact that when HNS becomes a monomer, there is a certain region of this particular HNS that is exposed to this protease that is present within the cell, and this protease kind of chews up this particular monomer. So what happens is that when you have lower concentration of let me start here to start with, and then I'll go into the other part. So when you have this particular observation, which I showed you before, that when you have this guys lining up the DNA and preventing transcription, and when you have a temperature which is 310 Kelvin, what happens is that this disassembles. When this disassembles, there is a protease that kind of eats up these monomers or chews up this, and hence you cannot form this oligomerization that can happen. But it's not as simple as that. And what we identified was that when you actually look at the monomer populations as a function of Anixton condition, they have a certain dependence, and they still have a significant fraction of monomers at around 310 Kelvin or so. But when you have an increase in monomer population, what happens is that when the protease kind of chews up some of the monomers because of the so-called Le Chatelier effect, where you have the equilibrium that is going to shift to more monomer population, there will be a chain reaction that will be set up, and there will be the overall concentration decreases, and hence the monomer population continuously decreases. So effectively, as you change the temperature, not only does the monomer get chewed up, what happens is that all the monomers in solution will be completely lost, and hence the oligomer will be disassembled completely, and even more monomers will be produced, and these monomers will also be chewed up with time. And so it actually sets up a very interesting chain reaction within the bacterial cell, right? So and this is very interesting observation, and we have just published this, so I haven't given this information here. All of these works are currently published in Nucleic Acids Research, and you can actually look at it. Right, so now you have a difference in the oligomeric status depending upon different conditions, which is temperature, solvent, ionic strength, or even the concentration. But the question is, does it affect the binding to DNA? There is not no prior evidence to this, and we actually went about doing simple experiments where you basically take a piece of DNA, you tag it with a dye, and you look at the rotational correlation time of this particular dye as you add more and more protein on it. As the protein binds the DNA, the rotation will be slowed down, and hence you will have an increase in anisotropy as a function of concentration, and this can be used to quantify some kind of binding affinity of this protein to DNA. And what you see here is that at 298 Kelvin, as you add more and more molecules of HNS, the anisotropy increases, which means that more DNA that is binding the protein, and it kind of saturates beyond this point. At 310 Kelvin, what you can see is that this curve is shifted to the right, which means at higher temperature there is weaker binding to DNA. And the interesting thing is there is no complete saturation. Ideally, you will see this kind of flattening out here, but it seems to continuously increase, suggesting that there is a specific binding, and this is something that is happening which is non-specific. So if you think that this phosphomimetic mutation is not oligomerizing, the question is, does it still bind DNA? And what we find is that, I'll just go to the next part, which is the ionic strength dependence, and also I think I should have I'm sorry, I thought I had it in this slide. But what you see here is that when this is at 150 millimolar, when you add or increase the ionic strength of the solution, despite the fact that you have higher order oligomers that are present, which means at the 300 millimolar ionic strength condition, we found that the octamer is the most stable state in solution. Despite that, 300 millimolar, the octamer is unable to bind the DNA because of the screening of electrostatics between the protein and the DNA, and hence you have weaker binding that is happening out here. So effectively what you see here is that the higher ionic strength eliminates the specific mode, which is this shoulder that you see here, which is not present here in this particular range of concentrations, and it's only the non-specific binding that seems to be present, right? So we still are trying to understand at a molecular level as to what is going on. And so what we can clearly say is that the DNA binding affinity weakens with temperature and also with increasing osmolarity. This is something that is known, but what is more interesting is that when you have the DBD alone, which means the DNA binding domain alone of this particular protein, and titrate it on DNA, what we find is that DNA binding domain is unable to bind the DNA. So by definition, a DNA binding domain is supposed to bind DNA, but unfortunately we found that there's nothing that is going on here. We can see it's completely flat, while the full length protein is able to bind the DNA very well. But now when we introduce this Y61E mutation, which means it's a phosphomimetic mutant that has only a dimer, this dimer is unable to bind DNA or binds only very weakly to the DNA, indicating that oligomerization is a prerequisite for DNA binding, which is the first of its kind that we have observed. And the reason why we think this is happening is because when the protein oligomerizes, this is a surface electrostatic potential map, what happens is that there is this large positive potential that is generated on one side of this particular protein phase. So which means that this guy is potentially able to enable stronger binding to the DNA, while when it's a dimer, this electrostatic potential is on the lower side, and hence it is unable to bind the DNA. That's the expectation that we have, and the question will come to phase separation. I will skip this particular part in the interest of time. Right, so I'll skip this part. So now I did say that I'll discuss about condensates and HNS, and I believe there has been some information on condensates from a couple of people at least, and I'll just give you a brief introduction to what these are. And this is something that has shot up, as I was discussing with one of you before, over the last 5-6 years in the field of biology. There are many different membraneless organelles that have been observed. And I mean, it has been known for a long time. The fact that these are membraneless and they have some interesting features associated with them is slowly being uncovered, and they are supposed to do a whole bunch of processes within the cell. They are supposed to enable rapid transcription, rapid translation. They're able to sequester antibiotics sometimes. Some of the drugs that we take in are also being able to sequestered within these particular condensates, and they can actually modulate folding as well. Some chaperones and enhanced enzymatic activity can also take place within this condensates. And the condensates, as you know, as just discussed in the last talk, you can have a series of interactions just like any molecular process. You can have all these charge dipole, cation pi, van der Waals interactions. All of these can play a role. On top of which, you also have an interplay between structural domains and low complexity regions. What I mean by that are those regions that are well folded and those regions which are disordered in solution. So the interplay between these two can also kind of regulate the degree of phase separated condensates that people find within the cell. On top of which you have something called multivalency. When you have repeated domains that are present in a particular structure, these can interact via heterotypic interactions and form condensates as well. All of these has been studied quite a bit over the last decade or so. And now, given from the perspective of HNS, it has well-structured domains. It also has low complexity sequence, meaning disordered regions which are enriched in charged residues, and it also forms oligomeric assemblies, and a certain phosphomimetic mutation is also eliminate these oligomeric assemblies to result in a dimer alone. The question is, does it phase separate with DNA? And right, so because it is supposed to condense, it's supposed to bind and condense DNA, we expected something of this kind to happen. And this is the structure, just to kind of recap. This is the structure of this particular protein. This is the disordered region, and you can see that this is nice little helix that is not interacting anywhere. And this is the DNA binding domain. This is the dimerization site, and this is the large electrostatic potential that you have on this particular protein. So what we did was just take certain concentration of the protein. In this case, I believe we took about 50 micromolar of protein and just about 400 nanomolar of DNA, and you put these guys together. This is DIC image, and maybe you don't see it well. This is a fluorescently labeled protein, and you see this nice droplets that are formed, and you can see this protein-rich condensates and protein-poor solution that you have here. So the protein is actually able to phase separate in the presence of DNA at a certain concentration. For the DNA binding studies, we used a concentration of DNA which is 1/10 of this, which we just used about 40 nanomolar. In this case, when you increase the concentration of the DNA, we see these nice condensates that are formed. And you can actually do something called fluorescence recovery after photo bleaching, and you can just zoom in on one droplet here, and you bleach this particular spot, and you can see that it's recovering with respect to time. This is some time axis out here, and you can see that there is completely bleached, and it slowly recovers because of the diffusion of the molecules within it. And hence, you call this fluorescence recovery after photo bleaching, and it's called FRAP. And you can see that with time, this recovers nicely to nearly 100%. Indicating that these are actually liquid-like. They are not solid condensates, they are actually liquid-like in the interior. And you can also see this nice fusion events that take place within a matter of few seconds. You can see this droplet that is kind of coming towards this larger droplet, and it kind of fuses, and then it fuses even more so. And there is also a nice concentration dependence that we see here. So at low concentrations of the protein, and it's about 1 micromolar, and you see no condensates formation. But as you increase the concentration, you start seeing these droplets. In all of these experiments, the DNA concentration is fixed to 400 nanomolar. And you can see that as you increase the concentration of the protein, you start seeing bigger droplets, more droplets, and you can see a lot of weird shapes as well, because these droplets are actually wetting the surface on which we are doing these experiments, right? So, and the interesting thing about these droplets is that they are very, very metastable. They are not just present there and you can see them for hours on end. No, it is not the case. You can see here the time dependence of the measure of droplet formation. This is turbidity at 350 nanometers, which is basically a scattering experiment. And you can see that there is some kind of higher turbidity at lower time points. But as you increase or you look at it longer and longer, and what you see here at around 5 hours, the turbidity decreases completely. And you have something that happens later, which we are trying to understand. We think that there is some kind of a liquid to solid transition that is happening in this particular case. But the interesting part is this particular region where this full length protein oligomerizes, forms condensates, and is forming condensates which as a function of time, right? These are metastable. But this Y61E, which does not bind DNA, is also unable to form droplets. As you can see, there is no increase in OD in this particular range of temperatures. And the question now is oligomerization really a prerequisite? And you can actually, this is just the same data that I'm showing you here as a function of time. You can see that the 0th minute you see these droplets. As you increase temperature, the time points at which you observe, and longer and longer times the number of droplets actually decrease. So basically the droplet kind of dissolves, and it probably forms aggregates. There's some issue here with the data which we are unable to completely understand because we are unable to see the aggregates. My student is currently working on this, trying to figure out what is going on. Right, so the next question that I wanted to ask is, now we found that This is HNS is a nuclear condensing protein, and now if you have changes in temperature and you go from low temperature to high temperature, and we already know from the thermodynamic modeling that the protein de-oligomerizes at higher temperature, and which means that when it de-oligomerizes, it should not be able to form droplets as well. And this is exactly what we observed here. This is OD as a function of temperature. Focus on this particular blue curve. As you increase the temperature, what you see here is that the OD actually decreases significantly. OD more the optical, basically this optical density as we call this, or turbidity, more will be the droplet in this particular representation. But now as you go to higher temperature, there is almost zero droplet formation, which means that at 310 Kelvin, it is unable to form droplets, or the number of droplets that you see are on the lower side, though we are unable to do microscopy at this particular temperature. And this is not due to the melting of DNA. On this open circles tell you what is the melting profile of DNA. So there is no change in the structure of DNA in this particular range of temperature. So the DNA structure is completely unaltered when you change temperature, but it's the oligomerization status that changes, and because of the loss in oligomerization status, there is no droplet formation. And this is exactly what we predicted from the thermodynamic model, and this is the octamer population as a function of temperature, and it has a profile that is very, very similar to that of the optical density as a function of temperature, indicating that oligomerization helps in assembly or binding to DNA, and this binding to DNA also enables droplet or condensate formation. And the moment you have this phosphomimetic mutation, there is no higher order oligomerization, only dimerization. But when there is no higher order oligomerization, it not only is unable to bind DNA, it's also unable to form droplets. Right? So the summary, though there are many parts that I skipped simply because of the interest of time, what we have shown is that there is the assembly, or the ways in which HNS can sense environment can be divided into multiple ways in which this can happen. One is the fact that there is a folding-unfolding equilibrium between the folded HNS and the unfolded HNS molecule, and you can have oligomerization or dimerization, and phosphorylation can actually modulate this a degree of oligomerization that you have in the ensemble, and depending upon whether specific or non-specific DNA binding modes can result in very different assemblies. And there is also this co-repressor that can actually bind here and hence also regulate the binding to DNA, and this co-repressor also modulates the degree of ensemble heterogeneity. And we were able to show all of this, and the condensate for is not published yet, but some of this is already work done. And this work was primarily done by Binsi Lucas in my lab. She's about to graduate in the next few months, and the thermodynamic modeling was done by Mohammed Fait, and the analytical ultracentrifugation data was generated with the help of Susumu Uchiyama in Japan. And I thank all these funding agencies, though all of these rejected the grant, but I'm using the grants from this and running it somehow. Right. Thank you.
Okay, so we'll take a couple of questions.
Sorry.
So I have very nice talk. So I just wondered that why you only have a dimer, tetramer, and octamer. You don't have pentamer, hexamer, or so.
Yeah, it looks like it's that's the observation that we have, and to the extent that we could model it, and looks like these oligomers might still be present, because if I can go back, it's probably easier. Yeah. Right. So if you see here, you can see some density out here, some density out here, some density out here. Maybe there are these assemblies as well, assembly modes as well, but the populations are on the lower side. We wouldn't be able to quantify them to the extent that we can. But what you can clearly say is that these are the modes that are primarily available. And maybe when you have these modes, other modes which are trimer and pentamer and hexamer, they are probably very weak and in terms of their populations. And why that is? Because dimer is the most prominent structure, and hence the dimer forms a tetramer, and the tetramer is the next most populated structure, and two tetramers come together and form an octamer, and that's what we think is happening.
And does this kind of discrete distribution affect your droplet sizes?
No, we do not know what is happening within the droplet yet. All we see is that when you have the wild type with all the possible modes that are present, it does form a droplet. When you change temperature, when you increase temperature, you melt the oligomeric assemblies, and when you melt the oligomeric assemblies, you probably have monomer and a dimer, and we know that when the dimer does not bind the DNA, and hence it does not form droplets.
So I have a question regarding the first part of the talk.
Yeah.
So you showed that there is two sites for oligomerization. One is dimerization and one is this oligomerization site.
Yeah.
So my question is, how do they differ in terms of their amino acid composition? And when there is two chains for HNS, they can potentially bind to both these sites. So which of the site is more preferred?
Meaning, this amino acid composition is very different. I cannot say what exactly is happening here or the sequence composition of this right out of my head. But what you have here is that this is actually a partially structured region, and which is if you can actually purify this particular region alone, and we find that it's actually intrinsically disordered in solution. And when it forms this nice oligomeric assembly, it is actually being more stable. And this is the region where it dimerizes. But now you can see that this oligomerization site is available for higher order oligomerization. And hence, another couple of protein molecules can sit on top of this. And hence, you have this tetramer that is formed.
Hi. Does the HNS phase separate by itself, or it requires?
Yeah, so there is a question which I don't know whether I have an answer, simply because we have had some weird observations with this particular system. When we keep the HNS in solution for longer times, we do see something that is going on, and we see something like fibers, meaning very long fibers that is probably forming, and it might be because of all these guys coming together and forming a big linear structure that is coming together. That is a possibility, but I don't have any confirmation that that is what is really happening. And that is simply because what happens is that when you change conditions slightly, and we don't see it at some point. So I don't understand why this is the case, and there is a lot of sensitivity to the way you prepare. Sometimes you mix it slightly extra or a bit more, and you don't see these fibers anymore. So there's a lot of sensitivity to the way you prepare the sample. So we are trying to fully address this, but we do see that there is a time dependence. You take HNS solution and leave it aside for some time, the turbidity increases. So the turbidity increases with time. And of course, this is not a phenomena that is specific only to HNS. We know that most proteins, when you leave them for longer in solution, they will either aggregate, oligomerize, do all kinds of stuff. So all of these are metastable, likely many of these states. The question is, how relevant it is to the bacterial system would be the more interesting question, and we don't have an answer for it yet.
As a continuation, so the HNS inside the bacteria has other partner proteins with which it
Yes.
completes the DNA binding sequence.
Yes, exactly.
So if you add some of those proteins in, let's say, slightly lower concentrations, can it inhibit this fibril formation? Because in biological situations, fibrils may not be an issue.
Yes. So yes and no in some ways. So the HNS is not the only protein, as you say, in bacteria, but you also have this paralog, which is called STPA, which is a homolog of HNS, which is also present in the same enterobacteria, which has the exact same architecture of structural architecture of HNS, but it has a slightly different sequence. We were unable to purify this, and we are still trying to work this out. But the interesting thing that I can tell you is that when you take HNS wild type and the phosphorylated version of this particular protein, and you put them together, the condensates dissolve. So you take them in 1:1 molar ratio and you put them together, we don't see the condensates anymore. So the phosphorylated protein is able to kind of equilibrate with the already formed condensates, is able to go inside, kind of solubilize it by weakening the DNA interactions.
Yeah, I have one question. So in one of your slides, I think you showed that the droplet is basically dissolving with time. So, and you mentioned it's possibly due to some aggregation that is happening, right?
Yeah.
So is it possible, like, you do something like TEM to actually figure out if you are forming aggregates?
Yes. So we actually can use not just TEM, but even use THT fluorescence, which is a molecule that is sensitive to aggregates, and see what happens. But when you do it with the DNA, the THT is actually intercalating with the DNA and is giving artificial fluorescence. So the only way to address this question is to do some kind of TEM or SEM microscopy, and which we are planning to do this. Yes. We haven't done it yet.
So related question, just one second. So is it meaningful actually to look into the density of these droplets, these condensations, as a function of time?
Density in the sense the OD.
These small droplets, like condensates being formed, right? And you say that with time they dissolve.
Yes.
And so, is it meaningful actually to look into the density of these?
I mean, how do you measure the density of these droplets is something not clear to me. Of course, you can pellet it out, and you can measure the some concentration fraction or molar fraction of these guys that are in the pellet form or the condensed form versus the dilute phase. And that is the only thing that we can measure. But the density per se, you need to do single droplet analysis, and we don't have the setup here to do that. But it might be interesting. We did try time-dependent frap, but not for this particular system. Meaning, you take a droplet and you do a frap analysis, and you see how fast it recovers, and it tell you it's like liquid-like if it recovers fully. But now you look at another droplet, but like 30 minutes from the time you initiated the reaction, and you look at that particular droplet, you do the same frap experiment, you will see that it doesn't recover fully, meaning that there are some liquid to solid transition that has taken place, and hence the recovery is not complete. And that is something we see in another system, but not in this particular system. But in that case, we can actually make a connection as to how much it recovers.
And do you see, when you said aggregation, do you mean essentially a bigger droplets or something?
No. It basically aggregates within the droplets, and slowly you will start losing the droplet morphology, and you will start seeing these clumps of aggregates that you would see generally in proteins.
I see.
Okay.
Yeah.
Have you checked the conformation of this protein as it changes with the concentration with like a dimer or monomer or tetramer or something? And then this is with respect to the concentration. At the same time, you have done some temperature dependent studies also with respect to the dimer, tetramer and things. So as you know that, I mean, the protein unfolds, then it easily forms the beta sheet structures and then forms the pylons. So just question here is, I mean, have you checked that, I mean, the tetramer or maybe the octamer is retain the native structures?
In the final, because we haven't solved the structure of this particular protein by any experimental method.
The circular diagnosis will tell you that.
It will tell you only the degree of helicity, but for all the average ensemble helicity is what it's going to tell you. It is not going to tell you the helicity of each of the different states that are present. We will not be able to separate it out in solution and hence quantify them. The moment you take it out in some way or the other, then you will reduce the concentration, and hence it will re-equilibrate to another set of states. So
Iso-exclusion chromatography, you can separate the tetramer.
No, we see one continuous elution profile. So we do not know when the tetramer stops and the octamer starts, and so on and so forth. Yes, but even if you take it out in the octamer form and you solubilize it, what happens is that the concentration now is going to be lower compared to the original starting concentration. And hence that will now kind of re-equilibrate into a collection of monomer, dimer, tetramer again. So it will be a continuous process, so that's a problem. Unless we are able to separate it out and even solve the structure, that will be the ideal thing. Say cryo-EM or something would help here, at least for the higher order assemblies.
Similar to what you described about the aging effects of time-dependent frap. So how do you tell if it's a liquid or it is in some metastable state from the recovery profile?
All we can say, and that's the kind of standard in the field that I'm also getting used to, because the literature is quite crazy out there. Well, he should know more because we had this discussion for 2 days, is that the fact that when you have a recovery which is complete, when you photo bleach a certain region and the fluorescence completely recovers in that particular region, and which means that the molecules are able to freely move around, and they are not stuck in some kind of a glassy state or some kind of aggregated state where the recovery is extremely slow. Here, within a few seconds or within tens of seconds, we see complete recovery in the fluorescence.
But you're not seeing the spatial movement, correct? You're just looking at it at one position.
We are looking at one droplet, and you photo bleach within that particular droplet, and you're looking at the intensity profile within that particular region. That's all we have.
But you don't look at the diffusion of...
No, we don't look at the individual molecules moving back and forth. No, that's not possible. The single particle tracking that we cannot do here. No.
Good.
Okay, so I think we will move on to the coffee, but for that, let's stand here.